Hi everyone, my name is Koji Tanabe. I'm a founder and CEO at IPS Inc. So I started my scientific career in the Dr. Shinya Manaka's lab. I developed the critical methodology to create the world first human iPSC, and I'm leading the author of that paper. That's why basically I loved iPSCs and I want to bring down this technology to the bedside as soon as possible. And the IPS vision is a peace of mind of the iPSCs. So at IPS, we are committed to realize the world where the cell therapy is accessible to everyone. So we are strongly believe that cell product is the essential tools for the next generation medical cares. So we are an enabler of the cell therapy as a true global provider of the GMP cell product with the multiple lineup using our mass production capabilities. So, but as you know, the iPSC technology is a perfect tools for the regenerative medicine and the drug discoveries. So there are already a lot of iPSC application is ongoing. And however, there are a huge bottleneck in the field. So that is there are the limited access to the GMP grade iPSCs for the commercial use. So this is because the process is complex and a resource intensive process and requiring the specialized talent. And to use these iPSCs globally, and these iPSC should be compliant with the global regulations, which strict and also not standardized. So IPS is removing this bottleneck, and so the industry can accelerate. So IPS already have the FDA registered GMP facility. We have expanded the facility from 1 to 3 GMP suite this year. And also these facility and also documentation control is free compliant with the 21 CFR 210, 211, and the ICHQ 7, and also Japanese and EU guidelines as well. And also I think our resource of these technicians has a very strong background to create clinical grade iPSCs. And also IPS has a very strong technology to create the high quality of clinical grade iPSCs. And our protocols can produce the high purity induction of the iPSCs, even at the passage 0. So as you can see, even if without passaging, we can induce iPSCs without more than 90%. And also we have a great, I think, tools to introduce the reprogramming factors. And these, I think, reprogramming factors can wash out immediately without passaging. And also this year, we succeeded to make protocols to produce the clinical grade iPSC, not only from the non-T cells, but also from the T cells. So using these capabilities, we already established more than 10 lines of the GMP iPSCs, and we can provide the off-the-shelf, the couple lines off-the-shelf GMP lines and the corresponding research grade lines of these clinical grade cell lines. And also we are providing the iPSC custom manufacturing service, not only from the non-T cells, but also from the specific T cells. And recently, we succeeded to develop the algorithm to identify the specific HLA type from the genomic database. And now we identify the various type of the HLA, and we are making the iPSC with a specific type of the HLA under the GMP control. And what is the feature of our GMP iPSC lines? So our informed consent is free compliant, is approved by the US independent ILB for the commercial use. And also tissue acquisition process is compliant with the EU US and Japanese guidelines, and we are only use the GMP grade reagent and the media. And also our license process is very simple. And also our iPSC has a global FTO. And our technicians to produce the iPSC has a very strong background to create a GMP product. They are involved in the Japanese national project to generate the clinical grade iPSCs. And recently, we are building up the analytical testing lab that is ISO 17025 accredited for the iPSC specific assays. And also, I can also provide the custom assay development and validation as well. And so by the last year, I think IPS is only focusing on to produce or induce the iPSC and expand it for making the master cell stock and a working cell stock. But from this year, we are expanding our business scope, including a donor sourcing and also creating a custom iPSC derived differentiation product as well. And as I mentioned before, we already we have succeeded to make the algorithm to identify the donor, and we are doing the specific donor sourcing and then making the iPSC, and we are doing the CDMO. We are making the iPSC derived cell product as a CDMO to collaborate with our partners. And as an example, we are using these capabilities. We are collaborating with every therapeutics and Panacea and Peter, that we try to realize that these iPSC applications. As the finally, so I'd like to explain what is the next of the IPs. So one of the biggest promise of the iPSC technology is the autologous transplantation and regenerative medicine. We are firm believer of the autologous cell therapies. So IPs already have developed the system to generate the hundreds and thousands of the individual iPSC cell lines, and we will enable the autologous iPS derived cell therapies using these capabilities. And we are building the future, which everyone has their own iPSC when they are born, and then they can use these autologous regenerative medicine in their life. So thank you very much for your attention. And also, we are really happy to collaborate with the various companies. And if you can contact us through the website, it'll be great. Thank you very much for your attention.